Good morning. I'm Bobby, back with you. Welcome on behalf of New Age Cosmetic White, EdTech Healthcare Consultancy. I welcome all of you to the routine lectures on the subjects of biology and chemistry for those aspirants going for the All India Institute of Medical Sciences, NEET exams, and aspirants of the National Eligibility Test for the Council of Scientific and Industrial Research exams. We're on the subject of protein synthesis, and we're on the specific subject of proteolytic processing of insulin. So, going into the subject proper, we have the proteolytic processing of insulin. The figure shows that. Depicted here shows proinsulin and insulin, where you have a cleavage of a signal sequence, disulfide bond formation, and the removal of the connecting polypeptide. Right, and the figure depicts the proteolytic processing of insulin. The mature insulin molecule consists of two polypeptide chains A and B, joined by disulfide bonds. It is synthesized as a precursor polypeptide, proinsulin, containing an amino terminal signal sequence that is cleaved during transfer of the growing polypeptide chain to the endoplasmic reticulum. This cleavage yields a second precursor proinsulin, which is converted to insulin by further proteolysis, removing the internal connecting polypeptide. And going on to the subject matter proper, it is interesting to note that the proteins of many animal viruses are derived from the cleavage of larger precursors. One particularly important example of the role of proteolysis in virus replication is provided by HIV. In the replication of HIV, a virus-encoded protease cleaves precursor polypeptides to form the virus structure proteins. Because of its central role in virus replication, the HIV protease, in addition to reverse transcriptase, is an important target for the development of drugs used for treating AIDS. Indeed, such protease inhibitors are now among the most effective agents available for combating this disease. Glycosylation. Many proteins, particularly in eukaryotic cells, are modified by the addition of carbohydrates at a process called glycosylation. The proteins to which carbohydrate chains have been added, called glycoproteins, are usually secreted or localized to the cell surface, although some nuclear and cytosolic proteins are also glycosylated. The carbohydrate moieties of glycoproteins play important roles in protein folding in the endoplasmic reticulum and the targeting of proteins for delivery to the appropriate intracellular compartments and as recognition sites in cell-to-cell interactions. Glycoproteins are classified as either N-linked or O-linked depending on the site of attachment of the carbohydrate side chain, which is visible on the figure given down here. It's on figure 7.25. In N-linked glycoproteins, the carbohydrate is attached to the nitrogen atom in the side chain of asparagine. In O-linked glycoproteins, the oxygen atom in the side chain of serine or threonine is the site of carbohydrate attachment. The sugars directly attached to these positions are usually either N-acetylglucosamine or N-acetylgalactosamine, respectively. The linkage of carbohydrate side chains to glycoproteins is shown in the figure. The carbohydrate chains of N-linked glycoproteins are attached to asparagine. Those of O-linked glycoproteins are attached to either serine, shown on, or threonine. The sugars join to the amino acids. Right? And are usually either N-acetylglucosamine, N-linked, or N-acetylgalactosamine, it's O-linked, right? And most glycoproteins in eukaryotic cells are destined either for secretion or for incorporation into the plasma membrane. These proteins are usually transferred into the endoplasmic reticulum with the cleavage of a signal sequence while the translation is in progress. Glycosylation is also initiated in the endoplasmic reticulum before translation is complete. The first step is the transfer of a common oligosaccharide consisting of 14 sugar residues, 2 N-acetylglucosamine, 3 glucose, and 9 mannose, to an asparagine residue of the growing polypeptide chain. As seen on figure 7.26, the oligosaccharide is assembled within the endoplasmic reticulum on a lipid carrier, dolichol phosphate. So it is then transferred as an intact unit to an accepted asparagine ASN residue within the sequence ASN-X or ASN-X309, where X is an amino acid other than proline. Dolichol phosphate, this is the expression of dolichol phosphate. In fact, the illustration. The expression or the expansion or the definition of dolichol phosphate would be a lipid molecule and the endoplasmic reticulum upon which oligosaccharides are assembled for the glycosylation of proteins, right? So that's about a word expression there. It's been explained. As in the figure 7.26, synthesis of N-glucose glycoproteins. The first step in glycosylation is the addition of an oligosaccharide consisting of 14 sugars residues to a growing polypeptide chain in the endoplasmic reticulum, the oligosaccharide which consists of 2 N-acetylglucosamine or more. In further processing, the common N-linked oligosaccharide is modified, 3 glucose residues and 1 mannose are removed while the glycoprotein is in the endoplasmic reticulum. The oligosaccharide is then further modified in the Golgi apparatus to which glycoproteins are transferred from the endoplasmic reticulum. These modifications, which will be discussed later, include both the removal and addition of carbohydrate residues as the glycoprotein is transported through the compartments of the Golgi, as seen in figure 2.7. The N-linked oligosaccharides are different glycoproteins that are processed to different extents depending on both the enzymes present in different cells and on accessibility of the oligosaccharide to the enzymes that catalyze its modification. Glycoproteins with inaccessible oligosaccharides do not have new sugars added to them. Right? And the relatively simple oligosaccharides of these glycoproteins are called high mannose oligosaccharides because they contain a high proportion of mannose residues similar to the common oligosaccharide originally added in the endoplasmic reticulum. In contrast, glycoproteins with accessible oligosaccharides are processed more extensively, resulting in the formation of a variety of complex oligosaccharides. Figure 7.27 examples of N-linked oligosaccharides. Various oligosaccharides form from further modifications of the common 14-sugar unit initially added in the endoplasmic reticulum. In high mannose oligosaccharides, the glucose residues and some Alright, while I get those details for you, please take screenshots of these diagrams, right? Just do not forget to take screenshots. They're very important for you. You need to read them. You need to make a printout and make your files for the concerned subject. Make your records for references and future references and study, right? That's how you study a subject. And the glucose residues and some mannose residues are removed, but no other sugars are added in the synthesis of complex oligosaccharides. No mannose residues are removed and other sugars are added. Hybrid oligosaccharides are intermediate between high mannose and complex oligosaccharides. The structures shown are representative examples, right? And there we are in continuation. O-linked oligosaccharides are also added within the Golgi apparatus. In contrast to the N-linked oligosaccharides, O-linked oligosaccharides are formed by the addition of one sugar at a time and usually consist of only a few residues, as seen on figure 7.28. Many cytoplasmic and nuclear proteins, including a variety of transcription factors, are also modified by the addition of single O-linked N-acetylglucosamine residues catalyzed by a different enzyme system. However, the roles of carbohydrates and the function of these cytoplasmic and neoglycoproteins are not yet understood. In figure 7.28, we have the examples of O-linked oligosaccharides. O-linked oligosaccharides usually consist of only a few carbohydrate residues, which are added one sugar at a time. Attachment of lipids. Some proteins are eukaryotic cells and modified by the attachment of lipids to the polypeptide chain. Such modifications frequently target and anchor these proteins to the plasma membrane, with which the hydrophobic lipid is able to interact. These general types of lipid additions, N-myristylation, prenylation, and palmitylation, are common in eukaryotic proteins associated with the cytosolic phase of the plasma membrane. A fourth type of modification, the addition of glycolipids, plays an important role in anchoring some cell surface proteins to the extracellular phase of the plasma membrane. In some proteins, a fatty acid is attached to the amino terminus of the growing polypeptide chain during translation. In this process, called N-myristylation, which I'm going to elaborate right now, is myristic acid, which has the addition of myristic acid, a 14-carbon fatty acid, to the N-terminal glycine residue of a polypeptide chain. I repeat, the addition of myristic acid, a 14-carbon fatty acid, to the N-terminal glycine residue of a polypeptide chain, right? So that was explanation there, and is attached to an N-terminal glycine residue, as in figure 7.29. So I'm showing all these explanations, the explanation for the words and the figures, step by step, as I explain, is because there could be a lot of confusion when you listen to all these terms for the first time or are trying to understand the continuation of the lecture as I go on. So as I get all these details to you across anywhere in the world that you're listening to, please mention your doubts, queries, or any kind of clarification that you may need in this context in the comment section of the concerned lecture, and I'll be more than happy to send you the necessary explanations, right? And continuing now, the glycine is usually the second amino acid incorporated into the polypeptide chain. The initiated methionine is removed by protein lysis before fatty acid addition. Many proteins that are modified by N-myristylation are associated with the inner face of the plasma membrane, and the role of the fatty acid in this association has been clearly demonstrated by analysis of mutant proteins in which the N-terminal glycine is changed to an adenine. This substitution prevents myristylation and blocks the function of the mutant proteins by inhibiting their membrane association. 7.29 Figure 7.29 shows the addition of a fatty acid by N-myristylation. The initiating methionine is removed, leaving glycine at the N-terminus of the polypeptide chain. Myristic acid, a carbon fatty acid, a 14-carbon fatty acid, is then added. Lipids can also be attached to the side chains of cysteine, serine, and threonine residues. One important example of this type of modification is prenylation, in which specific types of lipids, prenyl groups, are attached to the sulfur atoms in the side chains of cysteine residues located near the C-terminus of the polypeptide chain, which is shown in figure 7.30. Many plasma membrane-associated proteins involved in the control of cell growth and differentiation are modified in this way, including the RAS-oncogene proteins, which are responsible for the uncontrolled growth of many human cancers, as seen in Chapter 15 of the concerned subject. That's protein folding and processing. Prenylation of these proteins proceeds by 3 steps. First, the prenyl group is added to a cysteine, located 3 amino acids from the carboxy terminus of the polypeptide chain. The prenyl groups added in this reaction are either farnesyl 15 carbons, as shown in figure 7.3, or geranyl 20 carbons. The amino acids following the cysteine residue are then removed, leaving cysteine at the carboxy terminus. Finally, a methyl group is added to the carboxyl group of the C-terminal cysteine residue. Figure 7.3 depicts the prenylation of a C-terminal cysteine residue. The type of prenylation shown affects RAS proteins and proteins of the nuclear envelope. Nucleolamines. These proteins terminate with a cysteine residue, cis, followed by 2 aliphatic amino acids, and more. Protein significant prenylation is indicated by the fact that mutations of the critical cysteine block the membrane association and function of RAS proteins. Because farnesylation is a relatively rare modification of cellular proteins, interest in this reaction has been stimulated by the possibility that inhibitors of the key enzyme farnesyltransferase might prove useful as drugs for the treatment of cancers that involve RAS proteins. Such inhibitors of farnesylation have been found to interfere with the growth of cancer cells in experimental models and are undergoing evaluation of the efficacy against human tumors in clinical trials. And the third type of fatty acid modification, palmitoylation. Palmitic acid, a 16-carbon fatty acid, is added to sulfur atoms of the side chain of internal cysteine residues, shown as figure 7.31 below. Like in myristoylation, and prenylation, palmitoylation plays an important role in the association of some proteins with the cytosolic phase of the plasma membrane. Palmitylation. Palmitate, a 16-carbon fatty acid, is added to the side chain of an internal cysteine residue. Finally, lipids linked to oligosaccharides like glycolipids are added to the C-terminal carboxyl group of some proteins, where they serve as anchors that attach the proteins to the external surface of the plasma membrane. Because the glycolipids attached to these proteins contain phosphatidylinositol, they are usually called glycosyl phosphatidylinositol or GPL anchors. As seen in figure 7.3, the oligosaccharide portions of GPI anchors are attached to the terminal carboxyl group of polypeptide chains. The inositol head group of phosphatidylinositol is in turn attached to the oligosaccharide, so the carbohydrate serves as a bridge between the protein and the fatty acid chains of the phospholipid. The GPI anchors are synthesized and added to proteins as a pre-assembled unit within the endoplasmic reticulum. The addition is accompanied by cleavage of a peptide consisting of about 20 amino acids from the C-terminus of the polypeptide chain. The modified protein is then transported to the cell surface, where the fatty acid chains of the GPI anchor mediate its attachment to the plasma membrane. 7.32 figure shows the structure of a GPI anchor. The GPI anchor attached to the C-terminus anchors the protein in the plasma membrane. The anchor is joined to the C-terminal amino acid by an ethanolamine, which is linked to an oligosaccharide that consists of mannose. Now, that would be the end of that particular lecture, and we'll be continuing on the next portion shortly. Right, we proceed from here with explanations on the various terminologies and processes of protein synthesis. Here it's titled, we try to explain transcription, translation, and protein synthesis, right? So, what do you remember what proteins are made of? Well, hundreds of amino acids linked together to make one protein. There are 20 types of amino acids, some we can make and some we can't. There are infinite combinations of amino acids, can be hundreds of thousands, monomers long. These long chains are called polypeptide chains. Just simple points to remember as you study. Protein synthesis. Well, protein synthesis is the process in which a cell makes protein based on the message contained within its DNA. However, DNA is only found in nucleus. Proteins are only made outside the nucleus in the cytoplasm. Now, protein synthesis. How do the many different messages within the DNA molecule get to the many ribosomes outside the nucleus? A molecular cousin of DNA, RNA, is used to carry these messages. Ribonucleic acids. The job of RNA ribonucleic acids is to carry messages from the DNA in the nucleus to the ribosomes in the cytoplasm. There are 3 types of RNAs. mRNA carries a message from the DNA to the cytoplasm. tRNA transports amino acids to the mRNA to make a protein. rRNA make up ribosomes which make protein. And ribonucleic acids is the next, is RNA. RNA is almost exactly like DNA except contains a ribose sugar instead of a deoxyribose sugar, hence the name. Contains uracil instead of thymine. RNA is single-stranded, not double-stranded usually. Ribonucleic acid, RNA, you see your cytosine, etc. These are the diagrams of those, right? AGs, ADCGs, and cytosine, right? Take these screenshots, it will be helpful to you. The nitrogen bases, the ribonucleic acid, the deoxyribonucleic acid, and the nitrogen bases. Protein synthesis occurs in 2 steps. That's transcription and translation. So transcription is basically the genetic information from a strand of DNA is copied into a strand of mRNA. Translation is the mRNA, with the help of the ribosome, forms a chain of amino acids, eventually forming a protein based on the information contained on the mRNA. The central dogma is, this order of events is called the central dogma of molecular biology. This represents that DNA, RNA, protein. Step 1, transcription. DNA unzips, enzyme splits apart base pairs and unwind the DNA double helix. Bases pair up. Free nucleotides in the cell find what they complementary bases along the new strands with the help of RNA polymerase. What will be different? Let's read on that, right? The new backbone formed. The sugar phosphate backbone is assembled to complete the RNA strand and separates from the DNA strand. Step 1, transcription. Now we're going to watch a small animation, right? And then we go in for the more complex animation. So let's watch this. So these help us to just note down these web addresses. You type them onto your address bar and you can watch them. If there's time after the lecture, we'll get into the transcription part of it, right? Let's just check. Okay, step 1 is transcription. We'll try it out. It says try it. What RNA strand will be made from the following DNA sequence? Right, step 1, transcription. What RNA strand will be made from the following DNA sequence? This is the RNA, right? Make a note. Step 1 and 1 by 2, RNA editing. An mRNA molecule has to be edited in order to be useful, so that there's a lot of unnecessary information that needs to be removed. An mRNA sequence that does not code for protein is called an intron. A sequence that is useful in making a protein is called exon. Step 1.5, RNA editing. DNA, transcription to pre-RNA in nucleus, exon 1, intron, exon 2, intron, exon 3. So the RNA editing happens here. It happens here, leading to an intron, and then to RNA in cytoplasm, exon 1, 2, and 3. Step 2, translation to decode or decipher the meaning of. Now that our mRNA molecule has been made, it's time for its message to be made into a protein sequence. So how does the mRNA sequence translate into an amino acid sequence? Step 2, there's translation. That's a problem. The problem is there are 20 different amino acids. There are 4 RNA bases. They are A, T, C, and G. Adenine, thymine, cytosine, guanine. Right? Step 2, translation. Please note this link and paste them onto your address bar, and you can see that animation, right? Now step 2, translation. So how do you exactly go about determining what protein your cells are going to make? First, divide the mRNA sequence into codons, as you just saw and heard on the animation series. Codons are 3 basis sections of mRNA, AUG or CUG, as you see here. Step 2, translation. Since each 3-letter combination codes for an amino acid, you need to figure out what amino acid matches up with each codon, right? So these are the genetic codes. Second letter, UG, AG, right? Phenine, leucine, serine, tyrosine. So watch these. So this is the first letter, second letter, third letter. U, and U is phenylalanine. UUC, UCU is, so you combine like that, right? So note this with the genetic code. Step 2, translation. Since each 3-letter combination codes for an amino acid, you need to figure out what amino acid matches up with each codon, right? You have the genetic code here. Try it out, right? Step 2 is translation. Since each 3-letter combination codes for an amino acid, you need to figure out what amino acid matches up with each codon. That's the genetic code. Make notes. Step 2, translation. Since each 3-letter combination codes for an amino acid, you need to figure out what amino acid matches up with each codon. Now recap. DNA is transcribed into mRNA in the nucleus. The mRNA leaves the nucleus and enters the cytoplasm. The protein is translated from the mRNA sequence using tRNA amino acids. We're continuing from there. We have protein synthesis, which has chapter number 13. But you don't have to note that, but it's about the subject matter proper. We have the following topics that will be covered. Overview of protein synthesis, proteins, chains of amino acids, and following. Approximately 2/3 of the organic matter of a typical cell consists of proteins, and proteins are the final products of gene expressions in most cases. And the term proteome refers to the complete set of proteins encoded by a genome, or the total number of proteins found in an organism at one point in time. They are made by translation messenger RNA, mRNA, using a molecular decoding machine known as ribosomes. So right now, all those terminologies and all those explanations will hold good as I proceed with this lecture, right? So while you listen, you can correlate with the terminologies that have been elaborated to you, right? And going on, translation refers to the conversion of genetic information in nucleic acid language to protein language. Proteins are polymers made of amino acids, and each of the 3 bases of DNA or RNA corresponds to a single amino acid in the protein chain. Not only is the genetic information carried to the ribosome by mRNA, but other RNA molecules take part in the process of translation itself. Ribosomal RNA catalyzes the formation of the peptide bonds between the individual amino acids, and transfer RNA, tRNA, act as an adapter between the nucleic acid and protein languages. because RNA lies at the very heart of gene expression to give proteins. Messenger RNA, mRNA, the type of RNA molecule that carries genetic information from the genes to the rest of the cell. Proteome, the total set of proteins encoded by a genome, or the total protein complement of an organism. Ribosomal RNA, rRNA, class of RNA molecule that makes a part of the structure of a ribosome. Translation, making a protein using the information provided by mRNA. Transfer RNA, tRNA, RNA molecules that carry amino acids to the ribosome. Now, in this chapter of protein synthesis, we have figure 1301, how many proteins per gene. Normally, each gene is transcribed given one mRNA, and this is translated into a single protein. Variations in the normal theme are B, alternative splicing, C, polyproteins, and D, multiple proteins due to the use of their different reading frames. One mRNA, one protein, multiple mRNAs, multiple proteins. So you have normal, alternative splicing, polyprotein, and frame shift. You know these things while you study the subject of protein synthesis. Now, overview. Each protein in every organism is made using the genetic information stored in the genome. The genetic information is transmitted in two stages. First, the information in the DNA is transcribed into mRNA. We just read that. Next, the information carried by the mRNA is ready to give the sequence of amino acids making up a polypeptide chain. This overall flow of information in biological cells from DNA to RNA to protein is known as the central dogma of molecular biological cells from DNA to RNA is known as the central dogma of molecular biology. So as seen in chapter 3 here, nucleic acids and proteins figure 3.20 and was formulated by Sir Francis Crick. So ribosomes use the information carried by mRNA to make proteins. The decoding of mRNA is carried out by the ribosome, which binds the mRNA and translates it with the tRNA. The ribosomes move along the mRNA reading the message, recruiting the proper tRNA at each step and synthesizing a new polypeptide chain. The correspondence between genes and mRNA dictates the way in which proteins are made. An early rule of molecular biology was Beadle and Tatum's dictum, one gene, one enzyme. So as seen in chapter 2, basic genetics. This rule was later broadened to include other proteins in addition to enzymes. Proteins are therefore referred to as gene products. However, it must be remembered that some RNA molecules such as tRNA, rRNA, and small nuclear RNA are never translated into protein and are therefore also gene products. Because, furthermore, instances are now known where one gene may encode multiple proteins, figure 1301 coming up. Two relative widespread cases of this are known, alternative splicing and polyproteins in eukaryotic cells. The coding sequences of genes are often interrupted by non-coding regions. The introns, these introns are removed by splicing at the level of mRNA. Alternative splicing schemes may generate multiple mRNAs, molecules, and therefore multiple proteins from the same gene. This is especially frequent in higher eukaryotes, in particular vertebrates, as seen on chapter 12. Regarding processing of RNA, this is part of the subject. A set of proteins generated in this manner shares much of the sequence and structure. So just reading these notes for you, gene products and end products of gene expression is usually a protein but includes various untranslated RNAs such as RNA, rRNA, tRNA, and snRNAs. Ribosome, the cell's machinery for making proteins, is called ribosome. So going to the next page. In eukaryotic cells, most mRNAs only carry information from a single gene and therefore can only be translated into a single protein. This causes problems for certain viruses that infect eukaryotic cells and that have RNA genomes, as seen in chapter 20 for viruses, viroids, and prions. To circumvent the problem, these viruses make a huge polyprotein from an extremely long coding sequence in their RNA. This polyprotein is then cut up into several smaller proteins. Finally, there are occasional oddities, such as the generation of two proteins on the same gene due to frame shifting, will be discussed later. Despite these exceptions, it is still generally true that most genes give rise to a single protein. Although there are exceptions, most genes give rise to a single protein. To help understand the very complex process of translation, this chapter is divided into different sections. First, the key components, genetic code, amino acids, tRNA, and ribosomes, which are described in detail. Then the process of translation is broken into 4 steps: initiation, elongation, termination, and ribosome recycling constitutes translation. Remember that. Let me repeat that for you. The process of translation is broken into 4 steps, namely initiation, elongation, termination, and ribosome recycling. The simpler bacterial process of translation is first explained, and then the more complex eukaryotic version is presented. Finally, protein synthesis in mitochondria and chloroplasts is discussed. The final part of the chapter deals with getting proteins to the current location in the cell and some of the post-translational modifications and amino acid substitutions that affect the protein's ultimate function. Proteins are chains of amino acids. Proteins consist of linear chains of monomers known as amino acids and are folded into a variety of complex 3D shapes. A chain of amino acids is called a polypeptide chain. So what is the difference between a polypeptide chain and a protein? Firstly, some proteins consist of more than one polypeptide chain, and secondly, many proteins contain additional components such as metal ions or small organic molecules known as cofactors, in addition to their polypeptide portion. That's a protein structure and function. 20 different amino acids are incorporated when polypeptide chains are synthesized. So strictly speaking, there are 22. But the other 2 are inserted only under special conditions to be discussed later. They all have a central carbon atom, the alpha carbon surrounded by an amino group, a carboxyl group, a hydrogen atom, and a side chain or R group, as shown in figure 13.2a. And to note here, proteins are linear polymers made from amino acids. Most proteins fall into complex 3D structures, right? And the simplest amino acid is glycine, in which the R group, is just a single hydrogen atom. In solution, under physiological conditions, the amino group and the carboxyl group of amino acids are both ionized to give a Switzer ion or Doppler ion, dipolar ion, with 1 positive and 1 negative charge. Amino acids are joined together by peptide bonds to give a polypeptide chain. The first amino acid in the chain retains its free amino NH2 group, and this end is therefore called the amino or N-terminus of the polypeptide chain. The last amino acid to be added is left with a free carboxyl COOH group, so this end is the carboxy or C-terminus. When synthesized, the polypeptide is elongated from the amino terminus toward the carboxy terminus. Apart from glycine, amino acids have 4 different chemical groups surrounding the alpha carbon atom. This is called the chiral or asymmetric center, as seen in figure 13.04. Consequently, such amino acids exist as 2 alternative mirror image isomers with different chirality or handedness. So let's just read out these terms for ourselves so we get to know the basics as we go, right? Alpha carbon with the sign there, central carbon atom of an amino acid that carries both the amino group and the carboxyl group. Amino acid is a monomer from which polypeptide chains are built. Amino or N-terminus, the end of a polypeptide chain that is made first and has a free amino group. Asymmetric center, carbon atom with 4 different groups attached. This results in optical isomerism. Carboxy or C-terminus, the end of a polypeptide chain that is made last and has a free carboxyl group. Chiral center, same as asymmetric center. Dipolar ion, same as Switzer ion, a molecule with both a positive and a negative charge. Glycine, the simplest amino acid. Peptide bond, a type of chemical linkage holding amino acids together in a polypeptide chain. Polypeptide chain, a polymer that consists of amino acids, the R group, any specified chemical group, in particular the side chain of an amino acid. And zwitterion is same as a dipole ion. A molecule with both a positive and a negative charge is called a zwitterion. So next is on chapter 13 is the next page on protein synthesis is a figure 1302, which we just read. General structure of amino acids. A generalized amino acid contains an alpha carbon atom, an R group, an NH2 group, and a COOH group. B. Glycine is the simplest amino acid with the H atom as the R group. Right? When glycine is placed in solution at neutral pH, it ionizes to form a zwitterion. Right? So note these points. These are very important. Take screenshots and make notes. Glycine in solution is a zwitterion having both charges. So amino acids occur as pairs of optical isomers. Natural proteins are made from the L-levoisomers. So in continuation with the former proper study subject, consequently such as amino acids exist as two alternative mirror image isomers with chirality, different chirality or handedness. A pair of mirror image isomers is known as enantiomers or optical isomers. Alright, pair of mirror image, L and D. So they refer to as the L and D form because solutions are chiral molecules. Rotate the planar polarization of light in either a left-handed L-levorotatory or right dextrorotatory direction. The amino acids found in proteins are all of the L form, although L amino acid are sometimes referred to as the natural isomers. D amino acids do exist in nature. The peptidoglycan that is found in bacterial cell walls contains several different D amino acids. Several peptide antibiotics also made by prokaryotes, example bacitracin, polymyxin B, and actinomycin D also contain D amino acids. The D isomers of amino acids are found in bacterial cell walls and some antibiotics. Right? So 2.1, 20 amino acids form biological polypeptides. The 20 amino acids found in proteins possess a variety of different chemical groups. Figure 13.05. This wide choice of possible monomers makes proteins very versatile with a wide range of properties and capabilities, including great variety of possible 3D structures. The amino acids may be classified into groups depending on the physical and chemical characteristics. The major division is between those with the hydrophilic water-loving and those with the hydrophobic water-hating R groups. Glycine has only a single hydrogen atom as its side chain, so it does not really fit into either group. The hydrophilic amino acids may be subdivided into basic, acidic, and neutral. Basic amino acids contribute a positive charge to the protein, whereas acid residues provide a negative charge. Strictly, this refers to the situation in solution. So before I can proceed to the next page, let me read out these important notes about the various terminologies and the crux of the subject. So, what are enantiomers? A pair of mirror image optical isomers, that is D and L isomers, are enantiomers. Hydrophilic means water-loving, readily dissolves in water. Hydrophobic, water-hating, repelled by water and dissolves in water only with great difficulty. L and D forms, the two isomeric forms of an optically active substance, also called L and D isomers. Optical isomers where the molecules differ only in their 3D arrangement and consequently affect the rotation of polarized light. Peptidoglycan, polymers that make up eubacterial cell walls, consist of long chains of sugar derivatives cross-linked at intervals with short chains of amino acids. Now a solution within let's go to the subject matter proper. As we go through all the details here, within the physiological range, neutral polar residues have side chains that are capable of forming hydrogen bonds. The side chains of the hydrophilic amino acids carry chemical groups that take part in the reactions. The active sites of enzymes, discussed later, often contain serine, histidine, and basic or acidic amino acids. So as we go back, just go back to have a look at the diagrams, right? So starting from here, the two generic amino acids are shown. The R groups R1 and R2 here represent the side chains of any of the two different of the 20 different amino acids that make up proteins. Okay, we look R1 here and the R2 here, right? And represent the side chains of any of the 20 different amino acids that make up proteins, the whole thing together. Each amino acid has an amino NH2 group. Where's the NH2 group here? NH2, this one. Okay, NH2, right? And carboxyl, COOH. COOH, right? COOH, this is the carboxyl group. This is the carboxyl group, right? B, a peptide bond is formed between one NH2 and one COOH group. Peptide bond is formed between one NH2 and one COOH, right? So the NH2 and these two together form a peptide bond, right? Not here. NH2 and carboxyl group, these two together form a peptide bond with water eliminated in the process. So when these become a bond, the water is released here, okay? Now, a peptide bond is formed between these two, the NH2 and COOH group, with water eliminated in the process. Dehydration synthesis. So this is called dehydration synthesis, which means it's a process in which during the combination of an amino group and a carboxyl group, a molecule of water is liberated, and that is known as dehydration synthesis. C. Successive amino acids are joined in a similar manner by peptide bonds to form a polypeptide. So very many polypeptide bonds together, peptide bonds together form a polypeptide bond, right? 1, 2, 3, so many molecules. And the polypeptide contains an amino, N-terminus, and a carboxyl, C-terminus. Okay, the polypeptide contains an N-terminus, and carboxyl, C-terminus, right? Peptide bond linkage. So this is the peptide bond. Okay, many of these together form a polypeptide. So this is the C-terminus, which can't contain the carboxy group. This is the N-terminus, right? So that makes it easier for you to understand. So formation of many peptide bonds. Right, figure 1304, the L and D forms of an amino acid. The four groups are arranged around the alpha carbon differently in the L form and the D form of an amino acid. Although they share the same molecule formula, one is the mirror image of the other. So you see this? L isomer and D isomer. Okay, so one is a mirror image. So these two together will form what? Enantiomers, yes. So general structure of amino acids. R, the H, the NH2 and COH. This whole thing is an amino acid, right? A simple amino acid which is hydrophobic. Hydrophobic is it doesn't like water, repels. Hydrophilic. Basic amino acids. So just like sociophobic and sociophilic, they easily merge into the society. So they're sociophilic. They don't merge with the general society. So they're sociophobic. So there's a lot of difference. Similarly, and hydrophobic, 20 amino acids found in proteins. Amino acids can be grouped by their physical and chemical properties. The R group for each amino acid is highlighted. Okay, going ahead with the subject matter proper. The neutral polar residues have side chains that are capable of forming hydrogen bonds. The side chains of the hydrophilic amino acids carry chemical groups that can take part in reactions. The active sites of enzymes, discussed later, often contain serine, histidine, and basic or acidic amino acids. The hydrophobic amino acids may be subdivided into those that are aliphatic: alanine, leucine, I think it's isoleucine, I'll have to check that, valine and methionine, and those containing aromatic rings: phenylalanine, I think that's the word, tryptophan, and tyrosine. These amino acids are largely structural in function, except for tyrosine, which has a hydroxyl group attached to its hydroxy-aromatic ring, and can therefore take part in a variety of reactions. The classification of tyrosine is ambiguous, as its hydroxyl group is polar in nature. Proline has a non-aromatic ring and contains a secondary amino group, NH, rather than a primary amino group, NH2, like other amino acids. Such structures are sometimes referred to as amino acids rather than amino acids rather than amino acids. What's that again? Proline has a non-aromatic ring and contains a secondary amino group, NH, rather than a primary amino group, NH2. Okay? So secondary amino and primary amino, there are differences. Secondary amino will be NH, primary is NH2, like the other amino acids. So such structures are sometimes referred to as amino acids rather than amino acids. So what's an amino acid? Structure, it's one that has a secondary amino group, NH, rather than a normal primary amino acid that has a NH2. So two of the hydrophobic amino acids, methionine and cysteine, contain sulfur when a polypeptide chain is first synthesized. Methionine is always the first amino acid, although it may be trimmed up later. Cysteine is important for 3D structure as it forms disulfide bonds. The free sulfide group of cysteine is highly reactive and is often used in enzyme active sites or to attach various chemical groups to proteins, as seen on chapter 14, protein structure and function. Right, now the 20 different amino acids normally found in proteins may be represented by a 3-letter and 1-letter abbreviations, table 1301. So we'll look at that quite soon or shortly. These mostly correspond to the first letters of the name, but since several amino acids start with the same letter of the alphabet, the others need a little imagination. So they are used especially when writing out protein sequences. The amides, asparagine and glutamine, are relatively unstable and break down easily into the corresponding acid, aspartame and glutamate. Consequently, some analysis do not distinguish the acids from the amides. The abbreviation ASX and GLX were invented for these ambiguous pairs, right? Those that have similar letters and cannot be deciphered, we use the abbreviations of ASX and GLX. Now, going to the next portion, decoding the genetic information. These are 20 aminos. There are 20 amino acids in proteins. Excuse me. But only 4 different bases in the mRNA. So one base of a nucleic acid cannot code for a single amino acid when making a protein. During translation, the bases of mRNA are read off in groups of 3, which are known as codons. Each codon represents a particular amino acid. 4 different bases give 64 possible groups of 3 bases. That is, there are 64 different codons in the genetic code. Because there are only 20 different amino acids, some are encoded by more than one codon. In addition, 3 of the codons are used for punctuation. Those are the stop codons that signal the end of a polypeptide. Chain 1306 shows natural genetic code. And before that, the side note says each amino acid in a protein is encoded by 3 bases in the DNA or RNA sequence. Now this lecture, once over, would be continued for the concerned subject, as we have a couple of chapters that describe the entire aspect of protein synthesis and aspects. So there'll be following lectures from henceforth for a few lectures from now on, right? So keep in touch, make notes, make screenshots, so you're up to date, right? This particular lecture will be finishing shortly in about another minute and a half, right? That's about an hour. So you can go in. So all the lectures you can see it's at 55, 44 or 45. That's the time frame scheduled by the concerned software here. So after that, we'll go in for the next lecture. So the maximum possible would be lectures at 55:44. There could be breakages in between, but there will be continuations because each one will be continued, right? So going to the matter proper, it's about just going to end up right now. So just finish a few points and then stop off. Now each codon represents a particular amino acid. 4 different bases give 64 possible groups of 3 bases, that is 64 different codons in the genetic code, because there are 20 different amino acids encoded by more than one codon. In addition, 3 of the codons are used for punctuation. Those are the stop codons that signal the end of a polypeptide chain. So this decoding the genetic information will be a continuation in the next lecture, okay? So to read the codons, tRNAs recognize the codon on the mRNA, right? At one end, each tRNA has an anticodon consisting of 3 bases that are complementary.